Hi, my name is Kelsey Springer, and I am a second year vet student and a summer scholar here at University of Minnesota. So this summer, I worked on investigating loss of function variants in the general equine population. We've seen massive successes in diagnosing and treating genetic diseases in humans based on those famous large-scale whole genome sequencing projects. Um, a surprise finding from those efforts was that many gene variants predicted to disrupt normal protein function or loss of function didn't actually cause disease. So this was in part due to inaccurate gene annotation and genotyping errors. And considering that many horse breeds are predisposed to genetic diseases, whole genome sequencing is as promising a tool in equine medicine as it is in human medicine currently. So, understanding what variants are truly detrimental will improve our ability to identify genetic disease in horses. Here, 5,020 loss of function variants were identified in over 500 genomes, and a selection of those were split into a commonly inherited population and a rarely inherited population. Two main goals were developed for this study to first remove variant errors from both populations, and second, to investigate the functional consequences of the true variants. So, um, just a quick overview of methodology. First, I would determine where the mutation was in the equine nucleotide sequence as well as the protein sequence. If it was not annotated, it was classified as false. If true, I analyzed the protein sequence um, for evolutionary conservation of the target amino acid and whether the mutated version had deleterious consequences on the protein function. So, after repeating that on uh, hundreds of variants, what did we find? Um, so, found that the rate of false loss of function variants in the equine population is around 23%, compared to just 11% in humans. This validates strong concerns that there were more genotyping errors in horse genomes than human genomes. More specifically, for common variants, only about 2 out of 5 were true, and only 1 out of 5 of those true variants were in the sweet spot for being both conserved across species and deleterious on protein function. Meaning that after all is said and done, only 6% of predicted common variants are candidates for further investigation for potentially causing genetic disease. Concerning rare variants, the overwhelming majority are true positives, and about two-fifths of the true positives are both deleterious and conserved, meaning so far about 35% of the predicted rare variants may be worth exploring further. This showed that common variants were significantly more likely to be false than rare variants. So, for the sake of time, I will not cover how specific types of mutations impacted these variants. But if interested, please get in touch with me or the Equine Genetic and Genomics Lab. Overall, this project was a small part of a broader effort in improving diagnosis of genetic disease in horses. Um, Many variants from this project are going to be investigated further with RNA-seq and phenotyping, hopefully this coming summer. So with that, I'd like to thank Dr. Molly McHugh and her postdoctoral fellow, Dr. Sean Deward Ackhurst, for hosting me in the Equine Genetics and Genomics Laboratory for this year's Summer Scholar Program. Thanks for listening, and please get in touch if there are any questions.